So last we were here, we talked about transpiration. Give me a minute, please. Okay, so we talked about transpiration, and now we're going to move on and see what else do we have. Like I told you, everything else that we have is related to the histology or the anatomy of the details of the cells. So we need to do that now, and a few of the processes, right? This is an illustration showing you a structure from which we've zoomed in. So this structure is essentially the root. We've zoomed into two cells of the endodermis just to show you the Casparian strip. So you can see that the symplastic pathway from within the cell goes unblocked, whereas the apoplastic pathway cannot, because there is a complete covering, a 360-degree strip of a substance called suberin. It's similar to the lignin, which is thick, tough. The cell makes it in this strip place only. And this is how the apoplastic pathway is blocked. So what happens is, as a cell grows older, what ends up happening is this suberin builds up so much that it completely surrounds the cell. And eventually, what happens is all of the pathways are blocked. So the older cells become completely blocked and no more water can pass through them. As a result, the life of the cell is over and now the cell can die, right? Um, and then when this happens, there are some cells that remain in the root that are called passage cells, through which symplastic pathways remain open. But you know, like everything has to die. This is one of the mechanisms of senescence in a plant. When all of its root cells will be blocked, it cannot take up water. The plant is really old, it's going to die, right? Or when there's so much toxicity in the soil, why do some plants die in certain weathers, or all plants die, cannot grow in winter? Because of the water. When they sense that the water is very less, other substances that are coming in, salt is coming in, for example, in a salty land, suberin, instead of a Casparian strip, it deposits 360-degree, and then the plant has to eventually die because no water can enter through this 360 deposition of Casparian strip. It's no longer a strip anymore, it's just a suberin covered cell. Okay, now the drawing part. Before moving on to the drawing part, if you remember from your O level, there were two definitions that you learned in plants, monocots and dicots. If you don't remember, now is the time to remember it again. Monocots are plants that have a single cotyledons. Cotyledons are basically storage organelles that occur, organs that are in the seeds. So if it has a single storage organ in a seed, it's called a monocoton, a monocot, a cotyledon, and a dicotyledon has two of these cotyledons or storage, starch storage or amino acid storage organelles. There are other differences as well. For example, monocots have their flowers in multiples of 3. They can be 3, 6, 9, but they can never be, 5. They can never be 2. So it has to be in multiples of 3. However, dicots have multiples of 4s or 5s. So one thing to remember is that monocots and dicots are different in this way in the flowering petals. Another thing is that their leaves, you must remember, dicots have those beautiful long wide leaves, normal network of veins that you can see branching out, whereas monocots are the grass or the shrubbery that has long narrow leaf and parallel veins, no branching network of veins. What you need to know in A level is how their vascular bundles are arranged, because not all times you will get leaves and seeds and flowers that are apparently visible. Sometimes when you grow up, you'll realize that you get something called a stem or a root. Now, how do you tell a monocot apart from a dicot under the microscope? By looking at the stems or the roots. So that is visualized by looking at the vascular bundles. In dicots, in a simplistic arrangement, you can see that dicots have a very regular arrangement. The xylem and what are the vascular bundles? Xylem and the phloem, right? The xylem and phloem are arranged very regularly. Yellow one, if let's say xylem, it's all on the outside, and they occur in these bundles of these smaller circular bundles inside, surrounding the outer layer or the circular layer of the entire stem, whereas monocots have a random distribution. So that's going to help you. Let's go into the detail of that. Wait. Mm. Okay, so look over here. You can see a dicot and a monocot stem in detail. Monocot stem, the vascular bundle, whether it's the bigger, for instance, if I say the light purple one is phloem and the dark purple dotted, no, never mind. The light purple one is the xylem, and then a dark purple dotted one is the phloem, because it's the sieve plate of the phloem. Like I told you, sieve plates form in them, right? So if I tell you that it's like that, then look at how randomly distributed it is. It's very randomly distributed. It exists everywhere. It's on the outskirts. It's also in the inside. It's randomly scattered. There's no direction. Whereas in a dicot, there is a clear direction. It's arranged in these circles on the outer ring only. The inner ring is empty, has no vascular bundle, other stuff probably. The outer ring has vascular bundle in mini circles, and each circle has its own xylem that is on the inside of the circular bundle facing the inside, and then a phloem facing the outside. Okay, so just know for that for now. Ignore this word for now. Just know that this is random assortment, and this assortment you can easily recognize. And remember, xylem is always on the inside and phloem is always on the outside. So these two properties you need to remember. Now, the tougher part. These are the images that usually come in. These images, you should be able to recognize these images. It becomes very easy once you know about the random distribution of the vascular bundle in a monocot, but a systematic distribution in a dicot, right? And then you should be able to recognize where is the xylem and where is the phloem. For instance, if this is the pith or the central tissue, then I know these arrangements are definitely the vascular bundle, right? And this vascular bundle has the xylem on the inside, phloem on the outside, or more like phloem in the center, and the outermost is something he keeps calling sclerenchyma. I'm going to tell you what sclerenchyma is, but till then, just remember three-layered structure: xylem on the inside, phloem in the center, and the outermost is the sclerenchyma. Are you guys even understanding me? Can you guys hear me, understand me?
Yes, Miss.
Yes, teacher.
Yes, teacher.
Okay, so these now this is the whole bundle, right? And this bundle, how will you recognize which one is the phloem, xylem, and sclerenchyma? One, positions. And the second thing, the xylem is going to be something that is going to be empty in the center. So you can appreciate these emptiness in the center. These represent the xylems, and then whatever is following it is the phloem, and then whatever is following that is the sclerenchyma. So it's not that difficult if you understand the basic structure, and make sure that you understand this structure for the basic structure first. Okay, again, okay, so now let's move on and see the cross section of a leaf from a dicotyledon. And this is in your book, very important to understand. And I'm also going to use a few of these diagrams. Now, when I was teaching you topic 1, I also taught you one thing. If I ask you to make diagrams out of these, how will you make them? How will you sketch these diagrams? I told you you're not allowed to shade. I told you you're not allowed to make fancy dots and stuff like that. It's supposed to be very simple. For instance, if he asks me to replicate this one, all I'm asked to do is make this one layer, then the inner darker layer, and it should be, because I'm drawing on a screen, it's very difficult. These ends should be closed. These shadings shouldn't be dark. They should be fine lined, and then I should be able to make these circles. Now, will I draw the insides of these circles? No, not at all, because I don't need to. That's going to be extra detail. All I need to do is draw the basic structure. But for here, what I'm going to do is I'm going to make the epidermis like I'm required to, and then I'm going to make the exact outer location, right? Now in this one, I can give a characteristic inside phloem look. Okay, so this is wrong. This is basically the direction it occurs in, right? The xylem inside, the phloem in the center, and the outside, right? So you should be able to replicate these diagrams to an extent that they are replicated correctly. For instance, I drew this, I then drew these two lines. This is representing the xylem, this is representing the phloem, and this the sclerenchyma, the inner layer and the outer layer of the epidermis, and I make a whole circle just like this. I do not need to draw a line for this. You can draw a line for this to show that there's a change in substance, but you don't really need to, okay? Because there may or may not be a change in substance. And like I told you, shadings are not allowed. So this is a change in color and not really an organelle. So make sure you keep these things in mind. When laying out your drawings, okay? So let's see an example for that. Now look at this one. Now this one, this is a leaf cut in the center. So you can imagine this is the rib of the leaf, right? In the center you have the rib going through which, and you should know by this point that even these, when I was talking about these veins, these ribs or the veins, these veins are actually vascular bundles, right? Because what are veins? Vessels, just like in animals. So this, if I'd say in the middle of a leaf of a dicot is the vein, the thick vein. Whenever you hold it, it makes the two sides, the two flaps bend in the center, right? Bend a little bit. And if you break a leaf half and open, there's stuff oozing out from it as well. So that's obviously the vascular structure. So this must have the xylem and the phloem, and that's exactly how it is. Look at this. It has xylem and it has phloem, and then it has the regular arrangement. Remember that lower epidermis, upper epidermis. Look at the palisade tissues. This layer is entire palisade tissues. This layer air spaces and spongy tissues, all spongy tissues, palisade mesophyll. So if you can see, and this is the midrib. Midrib, you can imagine why he's calling it the midrib. It's in the middle. It's a rib. It's holding it in its position. So upper epidermis, palisade, spongy mesophills, air spaces, lower epidermis, and then in the center, the midrib is made up of the phloem and the xylem. Now, what is this? This deposition around the midrib is something called the parenchyma. You can see it over here. So over there, we saw a word called the sclerenchyma. Sclerenchyma was present over here. Now we've explored a new word, which is parenchyma. Sclerenchyma was in the stem, but the parenchyma is in the leaf. So let's see what that is. Before seeing it, let's see how, if you're given something as complicated as this, how can you sketch it? And this is how. See how simple he has made it. He made a line, he made a general outline, and then he made a line to show, a very thin line to show you the upper epidermis, and a thin line to show you the lower epidermis. Then he made a line for the palisade mesophyll on both sides, and no lines for spongy mesophyll. He assumes that you understand that this thin flap here and this thin flap here is obviously cuticle, upper epidermis, palisade, spongy, and you don't need to show air spaces. They're not really a quantity. You cannot quantify them, right? This is how you're supposed to make your drawings. Again, look at the things he's making. He's making a separate one for xylem, a separate one for phloem, and then he's just showing you a dotted line for the gradual change in consistency over here. Even if you don't make it, that's fine. You just make one drawing over here for the parenchyma and then one line over here for the other parenchyma, and you're good to go. For instance, xylem, phloem, and then this line is for one parenchyma and this line is for the other parenchyma, and that's as simple as that. So ensure that you use this diagram given in your book as the base for whatever drawings you want to make, okay? So this is the basic structure for the stem. Now, other than the stem, there is, and you should be able to recognize the stem and then the root. How do you differentiate the root? So in the root, it's exactly the opposite. So in those structures, in these structures, you had a pith in the center in the dicot, and the sides were demarcated by these our vascular bundles. Over here, you have in a dicot root, you need to remember these things. You have an X-shaped xylem center and a surrounding phloem. Now, surrounding that phloem is then structures, the usual structures that you have. There is a single layer of cells that regulates the ions that go in. We call it the pericycle. Peri because it's right outside of the phloem. It controls what goes into the phloem. It's going to be a protective layer for phloem cycle because it's a single round layer around it. And then the usual structures that form the root, the endodermis, the cortex, the root hair cell with its epidermis, like I taught you yesterday. This is the dicot root. The monocot root is exactly like the dicot stem. A pith in the center, xylem on the sides, right? Xylem is on the sides as a singular structure. The phloem forms a circle, like I told you over here, circular, completely filling circle around the pith and the xylem, the pericycle to regulate the phloem, and then the normal structure of an endodermis, a cortex, and an epidermis. So is this clear? Are these structures for the stem and the root of dicots and monocots differences clear?
Yes, teacher.
Okay, then you can be able to appreciate this. Look at the xylem, the X-shaped xylem in the center, the plus-shaped xylem, surrounded by the phloem, completely surrounded, then completely surrounded by the pericycle and the endodermis. And then, like I told you, pericycle and endodermis are one cell thick, and then the cortex can be a large number of cells, and then the epidermis one cell thick with the root hair cells sticking out. This is histologically shown. You can appreciate the plus or star shaped xylem, the surrounding phloem. This one is obviously the phloem. You can, how can I tell that? Because the cells look very different from these cells over here. These look like the cortex, and then this layer around it, the first different layer must be the pericycle, and then the outer layer must be the endodermis, right? And then this is the cortex. All the cells are similar. So very easy. Oh my God, there's a diagram that I can't see, but never mind. Let's go back, and this is what happens when you're extremely overworked and still working. Okay, so this is, again, you can appreciate the root hair cell, the cortex. Look at this, endodermis, pericycle. Then you can appreciate all the network of xylems. It's showing how water symplastically and apoplastically can travel. And then these yellow portions are the phloems, okay? Very easy, not something very difficult, right? Now you need to understand the three terms, parenchyma and sclerenchyma, and something called a parenchyma. Parenchyma, you've been studying since O-level. I'm sure you don't know what it really is. Parenchyma is just a living cell, okay? It occupies any space. It can be intracellular, intercellular. They have no specific shape. They can be specialized to form certain specific cells. For example, the dermis can be specialized into shapes, but otherwise they're called parenchyma. Now, the development of parenchyma into collenchyma and sclerenchyma is simply the deposition of lining around them, which is why you see them in plants and not in animals. So again, and as the deposition increases, the living nature decreases. For example, parenchyma has no deposition, so it's weak, cannot sustain very high pressures, but sclerenchyma, and it's highly living. Sclerenchyma is on the opposite spectrum. It is dead, highly deposited, not living, and it's very strong. Okay, and Kalenkama is in the center. It's living, but it also has a lot of deposition of, let's say, lignin and cellulose, and it's strong enough to support something. So the examples of parenchyma are generally the growing cell that can be specialized into collenchyma and then sclerenchyma. Sclerenchyma, you know, is xylem, highly deposited, specialized function, but dead. Collenchyma, on the other hand, are living cells that have some strength in them. These include the palisade, mesophyll, spongy mesophyll, the cortex, the pith, the epidermis, all of our specialized cells. Eventually, they come from the parenchyma. So why is this important? This is important because you need to understand that inside these cells, there must be parenchyma. There must be some kind of collenchyma as well. Why? Because there are some epidermis and everything inside. But outside of this phloem, there is highly protective cells that are not xylem. They are not for transport. They are just there for support and protection of the phloem layer. These are the sclerenchyma. Like I told you, the dead tissue with no intercellular space, stuck together, there for support. So these are protecting the vascular bundle from the outside. For instance, if an insect comes in and wants to suck out the nutrients from the phloem, he's going to insert his beak like thingy into the sclerenchyma. It's going to get stuck there and it won't reach the phloem, right? So that is the purpose of the sclerenchyma. On the other hand, this structure parenchyma, it has some functions. It's not dead, it's living. It does provide some sort of support, but it can help the palisades and the mesophils in their processes of it has chlorophyll, which is why the entire leaf is green, and it can help the other cells in photosynthesizing. So it's not totally useless, but it provides great support for these vascular bundles to stay upright. Vessels need to stay upright, and parenchyma and sclerenchyma are one way of keeping it that way. So are these three layers clear?
Yes, teacher.
Great. This is one of the toughest topic in this entire topic, understanding the sclerenchyma in these diagrams. If you understand these diagrams, if you can tell them apart, if you know, you need to go back and learn these things, that which one is the root, which one is the stem, what's the difference in arrangement of these cells and tissues, and that's it. If you know that, then the moment you see it on the exam, you're going to pick it and you're going to score well. For instance, again, some examples of drawing and structure. Without even reading, you can tell me that this is a dicot stem. Why? Because if look at this structure shows a xylem, probably a sclerenchyma, and probably a phloem, right? So and look at how beautifully he has labeled them. He's labeled the outside epidermis. He has labeled the pith with a single line, and then he's labeled the vascular bundles in their exact size. And these ones weren't clear in the image, so he chose to leave them. He did not force drawing them in. So he also made the proper vascular bundle. He made a clearly outlined xylem, no shading at all. And then a phloem, even if you don't make a cambium, that's okay, because you don't know what a cambium is at this point. And even if you don't know what fibers these are and know the phloem or the xylem, you can just label them fibers. If you know this is the sclerenchyma, then you can name it the sclerenchyma, and that would be great as well. But look at how beautifully and simplistically this is drawn. Again, let's look at this one. Now, this one, what does it show? This and again, look at this one. If you were drawing it, I'm 100% sure you would draw a line here to transition between these two, when you don't actually have to, because these aren't important structure. They're just the parenchymas, right? Different cells with intercellular spaces. Okay, again, now drawing this random one. Again, you know this is the outer layer is an epidermis, so needs a double layering. Then you see a structure in the center. Now this looks like a root to me. Let's see what root it is. This root has a structure in the center. It has air spaces inside the root. And you can appreciate the empty air spaces, and you can also draw them because they are different from the parenchyma. This cortex was completely cortex, so no difference had need to be made except for the pith or the empty space here, right, which was shown over here. But this one needs to be shown. The air spaces need to be shown. Then you need to draw the center. Again, the center, must have an endodermis, a pericyte layer, and then you can appreciate the xylem is forming some sort of a cross here. As soon as you see that, it should click in your mind, okay, this is a xylem with a cross. It must have phloem surrounding it, endodermis, pericyte surrounding it, and endodermis surrounding it, and you have your perfect diagram. And that's how you're supposed to attempt these questions. You're not supposed to attempt them from any kind of foresight. You're supposed to attempt them by hindsight. So is this clear?
Yes, teacher.
Yes.
Okay. Yeah. So I think we can be done by this topic by today. Yeah, we have very less remaining. So again, just to show you from your book, the phloem cell, like I told you, the sieve tubes, it's a living cell, very few mitochondria here, which is why it has a companion cell with plasmodesmata, with a lot of mitochondria, high number of rough endoplasmic reticulum and Golgi most probably. Why? Because there's a lot of protein synthesis and everything happening just to supply this sieve tube element. So this one companion cell has to take load of two cells, itself and this sieve tube element cell, the phloem cell, or whatever you'd like to call it. Okay. So very easy stuff. And now you need to understand the last topic that we have left. We talked about how water moves and how water moves into xylem and in and out. Now you need to know how food moves in the phloem in the multi-directional way. Like I told you, it can be moving up, it can be moving down, it can be moving up and down at the exact same time. Excuse me. Okay. So now I've shown you the very easy bundle. You can appreciate the xylem with the water that can only move upwards, right? And then the phloem. Now the phloem can move in any direction, right? But then one direction I need to show you that is definitely going to occur is movement towards the root. Why? Because the root itself cannot provide, see, anything above the root, above the ground has chlorophyll to make energy, right? It's a source of carbohydrates and amino acids for the plant, right? But the root is not. The root will definitely need food. So our generic understanding of mass flow should be that one thing is mandatory to occur: movement from the source or the leaf should occur to the root, and that's what we're going to explore. And the general stuff is very easy. Your source will provide lots of energy in the form of carbohydrates and amino acids. That is going to move into the companion cells. The companion cells are then going to move all of that into the phloem. When all of that moves into phloem, you can imagine how much the concentration becomes high. And because phloem and xylem are right next to each other, their cells obviously made up of cells. When the concentration is so high and two cells are next to each other, will water move into that cell or will it not? It will, right? So if this one cell has very high concentration of amino acids and glucose, a large amount of water will move into it from the nearby xylem because it occurs in bundles, like I told you. And as the amount of water keeps increasing here, the pressure and mass is so high that it is pushed forward, right? It can be pushed upwards or downwards in both directions. But what is happening over here? Top below or at the top, I told you this is happening simultaneously. At the bottom, what happens is the food substances at the bottom are removed by the companion cells and given to the root hair cells and metabolized. So the food is removed from here continuously. Now the osmolality, or the I told you, the concentration of substances here becomes very less. So all the water that was initially high over here will move out into the xylem, into the surrounding tissues, into the root, everywhere. As a result, there is a very low mass or low pressure of all of the substances here. This high pressure pushes substances down towards the low pressure, and a mass movement of substances takes place down a pressure gradient. Because these are mass of substances, we don't call it diffusion. That's limited to molecules and particles, so we call it mass flow, which means an entire mass is pushed down because of this difference in pressure that is occurring. Okay, so you need to understand this point to move on. Let's see what we have moving on. So this is a better illustration. You can see photosynthesis moves in sugar into the companion cell, into the phloem that attracts lots of water. The pressure here becomes so high that it moves into a direction with low pressure. This pressure is low here because all the sugar, when it's reabsorbed, what's left behind is the water, and this water then goes here and there, and the main area is the xylem because transpiration stream is continuously pulling water in the xylem up. So it gives it a very good direction for water to be absorbed into the xylem. And see, water and electricity are two things that will always move in a direction with least resistance. So over here, the resistance was high because of lots of water and substances. It chose to move downwards. But then over here, when the resistance was the resistance here was high as compared to the resistance here, water chose to move outside and be transpired up the stream. right? So it's a continuous process in which water is absorbed, taken up, moves in, moves down, then taken up, moves in. Some of it keeps moving out. So not all water is lost by transpiration. A lot of it is used up. In fact, that's why plants are 70 to 80% water, right? And so, so if you cut off, for instance, how does a flower die? What you're basically doing is in that flower, you're cutting off you're basically cutting off the storage, right? Or the usage of all of that, all of that sugar and everything else. Now, the sugars can't be used up here. As a result, when the sugar can't be used up, it's stuck here. So the pressure does not develop, and the roots cannot the lower part of the plant can no longer use, because pressure flow difference causes mass flow to occur. So now the food cannot move down. As a result, when you cut a leaf, cut a flower from the stem, the area furthest away from the flower starts withering slowly, and then the flower is going to start withering slowly, because no more minerals, no more water maintenance. No matter how many times you put it in a vase of water, the transpiration stream is there, obviously, because it's transpiring, the heat is causing it to transpire, but this pressure flow isn't developing properly, so it's not getting all of the things it's photosynthesizing, it's not being able to utilize it. So the next thing you need to understand is that there are three processes occurring here. Active transport occurs from the companion cell, when the movement of sugar takes place through the companion cell into the phloem, and similarly uptake of these back for usage or storage, whatever the fate might be. Otherwise, we have osmosis occurring, or a pressure flow or mass flow gradient occurring. Now, what is this exact process of the active transport? It's a very easy process if you want to understand it. It's very easy. How is it very easy? It's dependent on a few ions and a few ion pumps. Now, this is some new information, so make sure you're attentive. It's the last slide, we're almost done. And now, what happens is you have the companion cells. Every cell really has hydrogen ions. What happens is where the energy is used, the active transport basically occurs. The source cell uses a pump to with energy to actively transport hydrogen against the gradient from low concentration in the other part of the leaf into itself. And that raises the concentration of hydrogen ions inside the source cell. Once this is high, the source cell has two important protein pumps, a hydrogen pump, and then the second one is a co-transporter. The co-transporter is a transporter that needs hydrogen. It has two spaces for attachment. One of these spaces should be filled with hydrogen to open it, and the other is to be filled with sucrose or amino acid that will come and attach, and both of these will move into the companion cell. Now, from the companion cell, these can be moved toward down the concentration gradient into the phloem, because and then this can be moved down due to the pressure effect, right? So one thing is clear, that you need to understand how hydrogen is sucked up and then move, and this sucking up leads to high concentration here, which is why this high concentration will lead to facilitated diffusion down the concentration gradient into the companion cell. Because it's facilitated diffusion, it wants a protein channel, and the body and the plant body was so clever that what happened was, in this facilitated diffusion channel, it made sure that the hydrogen cannot be moved out without a sucrose or an amino acid attached to the protein pump, and that's how both move in. The starch, the sucrose or the amino acid, moves into the phloem, and the hydrogen that was moved here is sucked back into the source cell. As easy as that. And then the other steps are just movement of water and these sugars into the sieve tube, and then movement from high to low pressure, and then they are taken up by the sink cell. This is the source cell where everything's being produced. The sink cell is where it's used. It can be used for energy right away, or it can be stored, or can be converted into other substances, for instance starches and cellulose, because that also needs glucose for it. It can be converted into glucose. It can be combined with protein to form glycoprotein, glycolipid, whatever the cell needs. Okay. This is a zoom in of what's happening. You can see if this is the source cell and this is the companion cell. This hydrogen pump actively transporting hydrogen using ATP. You can see over here into this cell. When concentration of hydrogen is high over here, facilitated diffusion can occur through a co-transporter that needs a sucrose and amino acid to move it back into the cell. Sucrose and amino acid can move into the phloem, and hydrogen is actively pumped back into the companion cell. And that is it. We're done. Any questions?